everyone's here, we'll go ahead and get started. Thank you, Team BioCHIPS. We'll tell you how this is going to work, okay? You have quite a panel of experts here who you're going to start your presentation, and they're going to, based on their choice, either interrupt you rudely, interrupt you politely, or wait till you're finished to ask you questions as we go. Don't worry about this timeline and how many minutes you have. We'd much rather have a good discussion, all right? The way we'll go is our external experts, off-campus experts will go first, and then our on-campus experts will go second, and then our mentor will go next, and then I will ask some other type of questions I'm qualified to ask. And then you will answer them, and then we'll be done. But one lesson to take home from a session like this, maybe one of your first times you participated in something quite like this, but whenever you're in a professional setting and you are presenting technical information to a room full of people, you need to do a little bit of homework ahead of time, which you didn't have a chance now, but it will be easy, don't worry, to figure out who you're speaking to, who's in the room, what their basic level of understanding is, because you have to craft your presentation to match your audience, right? So to go overly formal in that process, we're going to go on this end of the room and I'll introduce ourselves to you. And then as you get started, I'm sure you will introduce yourselves to us. Okay? So I'll start right here. I'm Dr. Frank Cole. I'm Professor of Environmental Science and Technology on campus and Director of the Gemstone Program.
Hi everyone, I'm Professor Ryan Sokol. I'm in the Mechanical Engineering Department and an affiliate of the Bioengineering Department.
Everyone, I think I saw everyone before.
Okay.
My name is Xiaogong Luo. I'm an assistant professor at Catholic University of America down in DC, and 20 minutes from here.
Okay.
And I'm Bill Bentley. I'm former chair of the Bioengineering Department at the University of Maryland and mentor to BioCHIPS.
I'm Wu. I'm a second year PhD student in Dr. Bentley's lab.
Excellent. And everyone else you know, right? Okay, so you can go ahead. That's fine. Yeah, that's fine.
Sorry,
Okay.
I feel bad taking it.
All right. Okay, so BioCHIPS, whenever you're ready to go, get started.
Good evening, experts, Dr. Bentley and Dr. Cole. It is with my greatest pleasure
Wait.
that...
Does that mean that Dr. Bentley and Dr. Cole are not experts?
Very good point.
I just want to formally acknowledge
I got you.
In one instance, it's true.
Sorry, go ahead. I won't do that anymore.
It is with my greatest pleasure that I welcome you to Team BioCHIPS Thesis Proposal Defense. My name is Kimberly, and I will be presenting on behalf of Team BioCHIPS today, along with my fellow teammates Jessica, Veda, and Sam. Allow me to begin by introducing the problem with which we seek to address. The pharmaceutical industry is imperative to human well-being and healthcare. However, the drug testing process within the industry is greatly flawed. For example, there are great discrepancies between animal testing and human clinical trials in the drug testing process, and overall the process is extremely long and expensive. Team BioCHIPS seeks to address this issue by utilizing an organ on a chip, a device that simulates the working conditions of a specific organ system. Team BioCHIPS stands for biologically creating human systems integrated with programmed sensors. This describes the basic premise of our research, in which we seek to revolutionize the organ on a chip model by integrating sensors into the device. More specifically, an organ on a chip is a micro device which mimics the conditions, functions, and structures found within a specific organ system. They are traditionally made through soft lithography, but a relatively new practice, 3D printing, has emerged, in which we also seek to explore. We will seek to explore both of these options in our methodology, and we will discuss this more in detail later. There are various organs that can be modeled after in an organ on chip, such as the lung and heart. but we chose to model our organ on a chip after the gastrointestinal tract and therefore create the gut on a chip. We chose the gastrointestinal tract because it is an isolated system within the human body that does not require other fully functional organ system. In addition, the gastrointestinal tract is an important organ system for nutrient absorption and is therefore a targeted location for drug delivery. The following slide depicts images of various organs on the chips and their specific designs. If I may direct your attention to the bottom left image, that is an organ on a chip designed and modeled after the human brain, in which the researchers who created this model seek to determine whether certain drugs could pass through the blood-brain membrane. The top portion of that design is filled with brain cells, while the bottom portion is filled with bioengineered blood vessels. There is a thin membrane that separates both of these portions, in which that membrane represents the blood-brain barrier. If I may direct your attention to the bottom right image, that is an example of how cellular response can be modeled and quantifiably analyzed using an organ on a chip. I will now discuss the several research questions our team hopes to answer as we continue through our research. The first of these questions concerns the fabrication of the gut on a chip itself, in which we seek to determine whether soft lithography or 3D printing is a more efficient means. The second question that we hope to investigate asks how can sensors be integrated onto a gut on a chip model in order to maximize efficiency. The following questions focuses upon once we complete our working model. This question asks whether our model can be used to test various amounts of drugs, while our fourth question asks whether the sensors that we created can be integrated onto other organ on a chips. Using these research questions, we hope to test this hypothesis, in which we hope to analyze if sensors can be integrated onto the gut on a chip model, then treatments such as vancomycin can be tested and analyzed against the effects of diseases such as Clostridium difficile. I will now pass it on to Veda, in which she will explain the methodology in which we will create our gut on a chip.
Thank you, Kim. So in order to test our hypothesis, we have developed a 4-phase methodology and created 4 sub-teams in order to accomplish this. The first phase is divided into 3 parallel processes, and we hope to explore 2 different types of gut on a chip fabrication. In phase 1A, sub-team 1 will perform soft lithography fabrication, and in phase 1B, sub-team 2 will do 3D printing fabrication. In phase 1C, sub-team 3 will develop a sensor platform that can be used in assembly and integration of sensors into the model. In phase 2, sub-teams 1, 2, and 3 will work together in order to actually integrate the sensors into each of the models. In phases 3 and 4, sub-team 4 will work to test our gut on a chip model with the antibiotic vancomycin, first without the infection Clostridium difficile, and then with the infection C. diff. The first method of fabrication that we are using is soft lithography, and this is a fairly well-established method for creating microfluidic devices. The first step involves creating a mold using a thin silicon wafer, and members of sub-team 1 will be trained in the fab lab in order to do this. The next step involves pouring a polymer called PDMS into this mold and allowing it to cure to form the foundation of the gut on a chip. Then a chitosan membrane will be formed across 2 channels of different pHs that create a gradient. And this will allow separation of bacterial media on one side of the membrane and mammalian cell media on the other side of the membrane. Finally, we concede calcium ions and Keiko-2 cells in a calcium alginate hydrogel that is sandwiched between the chitosan membrane. These 2 images depict gut on a chip models that have been created using soft lithography already that show the 2 different pH channels that is divided by the chitosan membrane, and these 2 will be used to separate the 2 different types of media. The next method of fabrication we will be using is 3D printing. And this is a fairly novel approach to building microfluidic devices. And it starts with creating a CAD design of the model, and we will be using the software SolidWorks in order to design our gun-on-a-chip model and include both the structural components of the model as well as electronic components such as sensor wiring inputs that allow chip-to-computer connection for data analysis. We will then convert this CAD design into a stereolithography format that can be read by the NScript 3D printer, which is depicted in this image in action, creating just a simple light-up device. And this printer is ideal for our purposes because it can deposit up to 4 different types of materials, including metals that can be used to incorporate electrodes. In addition to printing the electronics, we will be printing a sacrificial material with our gun-on-a-chip, which when removed can reveal sub-micron patterns that are created by the NScript 3D printer. Since this is a very novel approach to creating organ-on-a-chip models, we will be revising and further developing this methodology as we progress in our research.
This is an overview for how we will be making our catechol cadhesin sensors. We will be using a gold electrode because gold is inert and has the ability to conduct electricity. Cadhesin will be electrodeposited onto the gold electrode by immersing the electrode in a cadhesin solution and then applying a current. This will serve as an assembly to add the alternating layers of alginated cadhesin by a layer assembly. Overall, the catechol cadhesin sensor will allow for a flow of electrons to amplify the signal, and we will be able to use it as a platform to insert our sensing mediators and specialize our sensors. The 2 special sensors that we'll be using are the oxygen sensor and the vancomycin sensor. For the oxygen sensor, the sensing mediator will be glucose oxidase, and that catalyzes the reaction from glucose and oxygen to gluconic acid and hydrogen peroxide. For the vancomycin sensor, we'll be using the mid-2000 vancomycin assay as the sensing mediator, and that's a redox-labeled vancomycin. This catalyzes the reaction from NAD+ to NADH. As shown in red, hydrogen peroxide and NADH are the electrochemical products of the reaction and allow the electrons to flow to the electrode and emits an output in a current. If you know the concentration of glucose, then you'll be able to detect the levels of oxygen and thus calibrate the oxygen sensor, which we'll be using. The disease that we'll be studying is Clostridium difficile, and this is a contagious and deadly disease that can be orally transmitted. It is caused by a bacterium that inflames the colon, and about 29 days patients die within 1 month of being diagnosed, and it is one of the leading microbial causes of healthcare-associated infections. Its corresponding treatment is vancomycin, and attacks the most severe forms of Clostridium difficile by binding to the alanine residues of the peptide chains. This disrupts the cell wall components and disintegrates the cell walls to fight against the Clostridium difficile. One problem with vancomycin is vancomycin resistance, and we'll be testing this with the disease in our gut-on-a-chip model, as Sam will now explain.
Thank you, Jessica. So the first phase of testing will consist of measurements of various chip parameters to ensure that our gut-on-a-chip model is indeed fully functional. First, using Keiko-2 cells that have been grown and cultivated in media for 2 to 3 days, we will measure the TEER, or transepithelial electrical resistance, to ensure that tight junctions between cells have been established. We will introduce a dye to one side of the membrane, and using photomicroscopy, we will image to see the presence of the dye on the other side. Then we will introduce bacteria from openbiome.org, which are designed to mimic the normal gut flora of the human gut. We will introduce those bacteria at low densities on the luminal side, and using bacterial plating methods, we will determine the viability of the bacteria, and using live dead assays, we will determine the viability of the Keiko-2 cells. After this, we will be testing the membrane to ensure that it is properly permeable, using 3 different size particles, which will be small green fluorescent molecules, red fluorescently labeled antibodies, and large polystyrene nanospheres. We will image with FITC and fluorescence microscopy to ensure that there is a negative correlation between increasing particle size and amount of fluorescence on the opposite side of the membrane. Furthermore, we will use cell gel testing to model the flow of fluids in our chip model. We will introduce modulator and reporter cells to one channel of the chip, while the other will be filled with a clear hydrogel. The channel will be allowed to flow at a rate of 0.11 microliters per minute, and we expect to see that it takes around 2 hours for the cells to be transported downstream. Finally, using bacterial stains and cell-cell signaling, we will introduce cells to our chip and monitor using fluorescence microscopy to ensure that the gel is stable and that there is no leakage. After the first 12 hours, we expect to see the cells outgrow their microenvironment and subsequently stabilize. After we have conducted these measurements, we will introduce the enzyme-labeled vancomycin using the EMIT 2000 vancomycin assay that Jessica mentioned earlier. And what that will do is the enzyme will be displaced by the regular vancomycin, which will allow for NADPH to be oxidized to NADP+, which will cause a current that we will measure using our sensor. After we have conducted this measurement, we will add the Clostridium difficile bacteria along with the vancomycin. Using the concentration of vancomycin added upstream at the chip, we will be able to use the concentration found downstream to determine what percentage of it was absorbed. And we expect to see about 25% absorption, which is what is commonly found in animal trials. We will monitor the increase in gut flora, recolonization, and decrease in Clostridium difficile infection using fluorescence microscopy and gram staining. I will now hand it over to Kimberly to conclude our presentation with our timeline and budget.
So the following chart depicted here is our team's intended timeline. As you can see, the various subteams will be working simultaneously in order to maximize both time and work efficiency. We are currently in the spring 2016 semester, which means that we hope to start our research as soon as possible, receive the necessary training to do so, or look into funds in order to support our research. The following chart depicts the materials and their respective costs that will be needed for our research. Many of the generic reagents will be provided by our mentor, Dr. Bentley, and were therefore not included onto our budget. The proposed total cost is around $2,600, which we hope to meet by fundraising through LaunchUMB or through research grants. As a quick summary of what our project is, because of the ineffective drug testing process in the pharmaceutical industry, both billions of dollars and years are wasted. As a result, our team seeks to create our own gut-on-a-model chip and integrate our own sensors via soft lithography or 3D printing. We will then use our models and test vancomycin and its impact on the effects of Clostridium difficile. Potential research ideas that could stem from our research includes creating a complete model of the human body as opposed to an isolated system. Furthermore, another research idea could be to look into different combinations of sensors and different organ-on-a-chips for various research purposes. And finally, since 3D printing is a relatively new practice, this research could contribute to further refining and improving the process. We are now prepared to answer any questions and listen to any comments and criticisms you may have. We would like to extend our utmost gratitude to our experts, Dr. Luo, Dr. Sokol, and Mr. Shang, in addition to our mentor, Dr. Bentley, for offering their support, insight, and guidance as we continue through our research. Thank you.
Very well done. So we will start asking questions. I suggest that the four of you make yourself comfortable. All questions can be handled by any team member or tag team any way you want. I also would suggest that someone or multiple people capture every comment, every suggestion that's made here. So you don't want any of this wisdom to go uncaptured. This is still recording, and how you handle the questions is part of the evaluation process. Okay. So we have a tradition of starting with our off-campus experts first. So that we're going to go there. So straight from Catholic University. Go right ahead.
Okay, yeah. Thank you guys. I think it's very impressive. To get I read through the proposals you guys put together. In the first part, literature reviews, it's very impressive. Like you have go quite intensive about the things. going in the field, then pretty much to the point. So it's great. Okay, and also I'm excited that you guys trying to follow what I was working on before, and then also kind of nervous how that going to be turned out, because it's like something you really can achieve within the limited time or not, or something maybe that because my fault I didn't make it clear. Okay, anyway, so there's a few questions. Mostly, the first one is a common, I guess. It's like maybe there's some confusion between soft lithography and biofabrication. So when we talk about soft lithography, normally it's about, do you guys, maybe you can, if you know a little bit more, can you help explain a little bit, and or I can help?
Let's hear what you know.
Well, what we know about soft lithography is usually they have a gold silicon mold, and then they pour PDMS and allow it to cure for some time in heated conditions, and then they remove the mold. And so we're trying to afterwards figure out a way to integrate electronic inputs and outputs to allow for sensors to be integrated.
Yep.
That's like the basic, I guess.
Yeah. So probably it's a title of subtitle in the test, also in the presentation. I think what you described is almost right. So the big difference is soft lithography is part of the device fabrication. And then when we talk about biofabrication, is where we try to integrate biopolymers into the device and really work towards whatever got on the chip system we want to create. So first you need photolithography to create the mold. Then you do the soft lithography use the technology you described. You replicate the PDMS mold and bond to glass slide, and that is done. The rest, when you talk about the membranes, you talk about put the cells together. Those are biofabrication. So it has three technology all integrated together. Maybe you can just differentiate a little bit more, so that people don't have confusion about soft lithography and biofabrication. Okay, and second question I had is like when you talk about the sensors, and then you also talk about the guard on the chip system. How you guys think about you know individually how that have been done, and then that what you're going to do? How do you integrate into the device so that you can make it as a system? It's got on the chip, and then you're going to do your test, I guess, on the chip as well, right? So how are you going to integrate them together?
So we are making the sensors separately as the first phase.
Sure.
They're being made out of the chip as like a separate entity.
Mhm.
But when they are integrated, they are made directly on the wall of the gastrointestinal tract. So like you start with the gold wafer on the
Mhm.
wall of the chip and then build on top of that. So when you're adding like the chitosan layers and the alginate layers, they're going to be flowed through the channels that are already on the chip.
Mhm. But right now your channels make up PDMS. So on the wall, if we need the electrodes, like it would normally done on the glass slide, the sidewall of the glass slide. And here, if it's PDMS, probably it's not easy to get the electrodes done, because that was done from the soft lithography process.
Yeah, it's like to clarify, so how, basically I think it ties into just an overall question of exactly how do you envision integrating the electronics? Is it in the PDMS, or is it in the glass, or is there a different method by which you intend to have some type of conductive materials within your device? I think that part might have been a little bit unclear.
I think it definitely changes things when we factor in 3D printing, because that was not part of our original design. So I think that'll be different from how we do it with the soft lithography. I think we envisioned it going into the PDMS. If I, yeah, that's right.
I see a lot of nods. So that,
Yes.
that's right.
Yeah. So PDMS is not good to get the electrodes, to be clear. There are people who have done that. They make it flexible, stretchable electrodes, but it's a lot more process than what you need to really make happen. I would say you can just do the first layer of photolithography process, made the electrode underneath, on the top of the glass slide, and then now you bond your PDMS channels on the top, and you should be able to do that. Okay, but then later we'll also need to consider a little bit more details, but probably when you do it, you will figure out which one to do first. To deposit the catalyst alternate to make it as a sensor first, or you do the cell culturing first, and that's something you can install later. Okay. So yeah, that's my main question. I will see.
You should really keep going, or do you have to do any more? Or
No, there are others, small typo or anything. It's okay in the proposal.
okay.
Go right ahead.
Yeah.
So I think some of the questions were, so it sounded like there were a couple different electronic measurements that you were trying to do. One of the questions I had was, so I think we talked about the transepithelial, the TER measurement versus, I think there was myosin was another electrical measurement maybe? Are there any differences in terms of the electrode design for those different applications? Or is it all kind of the same electronic architecture? So from my side, right, so I actually, despite my background, I'm actually not great with the electronic side. All of my stuff has been more on the fluidics side and the manufacturing side, not so much the electronics integration. So genuinely, I'm just interested in exactly, so I think there are challenges, from my side. I wasn't 100% sure exactly how the electronics would be integrated from the soft lithography perspective. I've also seen a lot of examples where typically the electronics are deposited on the glass layer and then the PDMS layer is then put on top. Or like a sandwich approach where you have completely open microfluidic channels, so there's no covering, and so you would do the glass with the electronics, the microfluidic channel, but it's open on top, and then another glass layer on top of that, if you really need electronics on top and on bottom. But I was wondering if you could kind of expand on that part, the electronics integration, and then if there's any differences between the two different measurements.
So who's our electronics guru?
Another way to put that question is that was subgroup 3.
Yeah.
Have you guys done that yet or not yet? The subgroups.
Well, me and Monil have been sort of working on the sensors, and I don't think we've quite looked into as in depth into the electronics as yet. So, can you repeat your question?
So, all right,
How the heck do you hook the wires up?
So the first question, it's really a two-part question, which is poor form on my part. You're really never supposed to ask a group a two-part question that's considered like, it's like bad form. So my apologies. So first question is just, how do you hook up the electronics? Or how do you plan to integrate that with your system?
I know when you came to our meeting that one time, when you talked to us about 3D printing, you said that we could integrate multiple materials, and so when we 3D print them, ideally we would like to print them so it would just kind of be integrated along with it, so the entire chip would just be made with the electronics. As for the soft lithography, I think we would have to, have spaces where we can input wires separately at first before we kind of figure out a way to do it so that there aren't wires coming out of it, if that makes sense.
Okay. I mean, I think more just as feedback. So it'll be, so certainly soft lithography, I think as I probably mentioned before, it's a relatively simple process. It is not a challenging process. I have had tons of undergraduate researchers who learn the entire process within the span of a month and can do it. It sounds really complex, but in practice, it is not that challenging. However, electronics integration is. The difference between learning how to mold PDMS and actually being able to integrate the electronic components in a way that you are getting repeatable results that can give you accurate testing will be a challenge. So I think one of the things that will be important is just trying to develop a really solid protocol, or just at least an overall plan in terms of integrating that part. Because that's one of the parts that I worry about depending on like the architecture that you're trying to get in terms of electronics. The second part of that question was differences in the electronics design depending on the application. So for TER, is that the same exact set of electronics that it's just you do something different, or is it that has to be designed one way, and then when you do the sensing component later for the I think it was the vancomycin sensor, is that a totally separate set of electronics? Are they both running in parallel, or is it like you do one just for one test to see, hey, look, we've grown, you know, a monolayer here. It really is a monolayer, look, we did this test, and now we're using this totally different device with a different electronic design for the next part.
I think there'll be two separate sets of electronics. I think the sensors will kind of like stay within the chip, and so we can, I guess, find a way to connect the chip to a computer for us to analyze data from the sensors. But for the tier measurements, I think we'll have a separate set of electronics that we can attach to the chip and figure out and visualize like whether the junctions are tightly established or not.
Cool.
Yeah.
I think that's good. So I think obviously I think the electronics design is probably one of the most challenging parts of this, which is obviously why many groups haven't been able to do that. I mean, I think if you take a look, or obviously you've taken a look in the literature, a lot of people kind of haven't gone the integrating electronics route. It's usually been more qualitative or taking samples afterwards kind of behavior. So I think that is one of the areas in which you can definitely advance the field. One of the things that I was hoping to see more of in the presentation was a little bit more of a summary of the problems with the current state of the art. So I've read the work from Ingber's group that had basically the lung on a chip just adapted for the gut. But I was wondering if there are other advancements since that people or researchers kind of keep moving in a certain direction, but there's still certain limitations there that your approach of integrating the sensors and so forth will vastly improve, or is it more just, you know, you want to try to see if you can integrate the sensors? Is there a genuine need for it?
I think the problem is that a lot of researchers can make these organs on chips, which is a great resource. However, these chips can't really be used for much else other than learning about the organ, which is great for that research, but I think that technology should be able to be applied for a more practical use, such as drug testing.
I think adding to that, I think in the actual organs, sometimes there are changes in the organ environment that happen when you introduce a drug or bacteria, as we are planning to. And I think the sensors allow monitoring of that just normal gut-on chips that have been built so far don't allow. So that allows kind of analysis of the effects of changes in the environment.
So I had my own answer for the question, but I think that's more towards it. So the idea of having continuous monitoring capabilities, that's one of the strengths of having integrated electronics or integrated sensors, where it's not, so some of my past work in kidney on a chip, literally someone has to go in there every couple hours and take a little bit of the fluid and then save it. There's nothing continuous about it. It's a ridiculous process. So certainly I think that's one area that if you were trying to build a case for what your advantage is over prior techniques, why you want to go through all the different processes to integrate the electronics, I think the continuous monitoring is a very strong sell from my point of view. I'm trying to remember, I had maybe one other question, but maybe I'll hold out.
We can come back around.
Yeah.
Right. Can I?
Yes.
Thank you. I'm just curious first. So you mentioned 3D printing of electronics. I'm just curious, like how do that? What material are you printing? What substrate you're using? I mean, yeah.
So the 3D printing is kind of a work in progress, but I think because the end script can print 4 different types of materials, I think we would probably integrate some kind of conductive material and maybe build input and output ports like when we make our CAD design of the chip. We're still kind of, I guess, figuring out how exactly the electronics will be printed with the rest of the chip.
Yeah.
That's a good question.
That's just my curiosity. So my question is, so you guys want to culture Keiko2 cell in your device, like, and you want to make this device like from using PDMS or other material, like where is there a specific place in your device that can be used to culture this Keiko2 cell, or you just culture it on the side wall or any place in the device designed to culture mammalian cell?
I think the goal was to culture them externally and then import them into the chip.
Then when you import a Keiko2 cell, do you want them to form one layer in your device, or like, so where is that monolayer going to be in your device? Okay, just think about it.
So how we're planning on doing it was having like the PDMS mold have like the channels built into it, and then we'll have like a separate like area that's on the chip that'll be kind of like, I guess, just surrounding the channels and like simulating the 3-dimensional structure of the gut, and so we'll be seating them kind of like around on the walls.
So you said there will be a scaffold.
Yeah, there will be scaffold.
So how do you guys make this scaffold? With, like, what method what method you gonna use to make that scaffold? Like 3D printing or soft lithography in general?
So we're going to be using like the 2 different methods. So like we said, the 3D printing part, it's kind of still a work in progress and hasn't, like, we're kind of working towards seeing what exactly we can do with that and how we can integrate the cells. But with the soft lithography method, like I said, we're planning on just kind of using the chitosan membrane and incorporating that in, and like seeding the Keiko2 cells like directly onto the device.
Can I interject?
Please do.
So where do you get a chitosan membrane?
So so we were planning on using the 2 solutions, like a chitosan solution and calcium alginate like buffer solution.
Right.
So then using those 2, we would put those into the device via the 2, I guess, like flow pathways, and then they would create a gradient, a pH gradient where the chitosan membrane would form.
Okay, good. So
Yes.
you get 2 flows coming in.
Yes.
Right, so this is what he was asking. So you
Oh.
you have 2 channels, and you're gonna have a membrane that grows down between the 2 channels.
Yes.
Yes.
So that's how you get your membrane to grow the Keiko2 cells on.
Yes.
That's where so you don't have to put them somewhere obtuse.
Right.
They
Yeah.
go right there, right?
Yeah.
But they don't really go right there unless you trap them in an alginate gel.
Yeah.
Yes.
Okay, and so it's a little bit, that's why Dr. Lowe is talking about biofabrication, because that's not at all part of the soft lithography or 3D printing.
Yeah.
Because you've got to get those cells in that localized spot, and it's sort of a membrane-like structure. And that's where that alginate comes in. So you have calcium on one side and cells on another side. It makes that hydrogel.
Yeah.
Just so I think you get it, but you got to be able to try to articulate that. And he's the one that invented all of that.
So I'm nervous. What is going to be working for you guys? Yeah, so basically, yeah, exactly as what Dr. Benny described. So you're going to use biofabrication to make this membrane. So after the channel is done, there's a done deal for soft lithography. That's very easy, as colleague said, but you can just get the device done and bound it on the glass, and you're ready to go whenever you're ready to do the biofabrication. Okay, when you do biofabrication, you bring the different solution together and then let the reaction between these biopolymers happen, and you get a membrane. And the next is, well, you're really going to assemble the cells. So if you assemble K2 cells, if you really want to make it work as a tight junction layer, probably the way to do it is like you can assemble a collagen with alginate or just collagen, some way figure out it's doable as well. Or you can have a alginate layer and then just coat with alginate or BSA on the surface along the catalytic membrane. Now you promote the cell to live there. Okay, and then when the cell live there, they grow healthily as long as you keep them carefully, feed them, and then they will grow as a tight junction. So that is more into the details. Need to figure out the sequence.
But you just learned about a very valuable resource.
Right, and then the other part about the gold and the electronics. This guy has put the gold on the sidewall of a channel. It's not a PDMS channel, right? Or is it PDMS? It's glass, right?
Glass.
The electrode.
Both.
You have it on a PDMS channel?
No, a glass.
Glass.
But the channel is made by PDMS and glass.
Got it. But in the end, to get it to like hook up to your computer, the whole beauty of this is that you have a little alligator clip. So you have an electrode in a sidewall of the channel and then a wire of gold that goes across to the side of the cover slip or the glass slide that you're using, and you clamp that with an alligator clip, and that gives you the conductivity so that you can hook that up to the computer. It's very simple. So Dr. Sokol's concern about the electronics is very real, but one of the things that also was developed here was the sidewall deposition of an electrode that then allows you to just put that on. So that's using lithography. Completely agree with you guys that the 3D printing domain for this is totally a blank slate, and anything you come up with is going to be publishable, and it's going to be a huge advance in my view for the field. So there's some motivation there.
You guys more questions?
I don't have anything. Yeah,
Okay.
I'm good.
Very good. You want to come back to some more technical questions? Did you save any like hand grenades to throw anything in?
I mean, I had comments on presentation style, but that's not.
Let's hear that. That's perfect.
Yeah.
So, so one of the things that I thought was a little, I mean, you know, so from my end, right, I don't actually know the format of this presentation, so there's only so much that I can say on it. But I did feel that it, you know, if it's possible for, you know, as you get more mature in your, like, I guess your process is becoming, you know, professional researchers and things like that. One of the things that's important is to kind of clearly articulate the outline of your story. So typically that kind of goes in a process where you explain basically, you know, the overall field, you know, why this area is important. And so I think you did a good job of that. You explained, I think you talked about, I mean, the first slide was pretty good. You talked about the problem in the area, specifically with respect to pharmaceutical testing. So I thought that was good. The next step that I was hoping to hear was a little bit more detail about the current state of the art. So typically you talk about the problem, you talk about how people currently attack this problem, and then usually that's followed by the next step, which is, but here are the limitations of the current state of the art. An ideal system would include this, 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 and this. We can't get there, but I think we can do this and this. And then you kind of break it down a little bit more where you start talking about your real approach, but ideally with a little bit more visual representation. One of the things that I did notice was there was a lot of exposition during your slides. And one of the things that's great about having a PowerPoint presentation rather than a speech like at graduation where someone just comes up there and just talks to you is that you have this slide that you can include movies, you can include animations, you can just include pictures or schematics of what it is that you're proposing. There were a number of times where I felt that the people who were presenting, you were forced to kind of try to explain these different shapes and these different architectures where a simple picture would have saved you a lot of time, and I think probably would have communicated exactly what you wanted to us. Even in terms of maybe where the electrodes would go or things like that. All of that was kind of, it's said, but it's not clearly communicated. And so you never want to have any time where you're doing a presentation and there's any kind of disconnect between you, the presenter, and the audience, really for any reason. The other thing that I think I would probably just mention is that, and it's possible that it's just very early in the process, but when you start talking about the processes that you'd be using, or just kind of what that would look like, or where the different cell types would go, I think there was a lot of technical, it's not jargon, but it's just technical content. It was a lot of words and vocabulary that might be challenging for people that aren't hard into this field. Like I understood parts of it because I've been kind of around this field, but there are definitely parts where I was like, oh my god, like what are they talking about? And so if I'm having a rough time, it's possible that other people that you start presenting to that are outside of this bubble might have a tougher time. And so trying to put yourself as much as you can in the position of an audience and then calibrating it towards that, I think would have been helpful for future presentations. Yeah.
Okay. Anybody want to add something to that? I have a couple questions for you. Your budget, which you went over, which matches the money you had in your written proposal. I mean, to me, this sounds like a very ambitious project to do for $2,500 or whatever you proposed. I did notice a lot of blue text in the proposal saying provided by the Mentor's Lab, provided by Mentor's Lab, provided by Mentor's Lab. So you're very fortunate in that fact that you're being underwritten. Okay. But you do have to still come up with some money. And you use the perfect phrase that every student group in your stage of the game uses is, we're going to find grant money. So we now have this wonderful thing that we've been very successful with Launch UMD, which is our crowdsourcing platform. Our gemstone groups have been phenomenally successful raising money through that. Last year we raised about $80,000 through it.
Really?
Yeah.
That's crazy.
Yeah, it's great. It's wonderful. I love it.
It comes from all over?
It's
Not just
crowdsourced, individual people, like you give $20, I'll give $20.
But
and all
are
that
they all people on the campus, or do you get people?
They hustle it. They put out, they put out there. Grandma sends in a check.
Grandma, yeah, right.
I recognize a lot of the names. I write thank you notes on donations, and I recognize a lot of the last names.
Yeah. That's fair.
But it's really good. I mean, so that's usually capped about $5,000. That can cover all your expenses. But you also mentioned the generic, we'll get other grants. Where do you think money might come from for a project like this? Whether you're going to pursue it or not, where would you, if you had to pursue it, where would you look?
Probably from like NIH and the FDA, because a lot of federal governments can also support like studies in this area. Really.
Okay. I'm sure there's some serious competition out there for those dollars. How and why would a group of sophomore undergraduates compete for that money?
So, I mean, I think that it's definitely worth a shot for us to try for grants from the NIH and NSF and such places, because I think the project that we're trying to do, even though, yeah, we are sophomore undergraduates, we are part of an esteemed program at an accredited university and in the Honors College, and also I think the project that we're trying to do is something that is definitely novel. And I think, like Dr. Billy had mentioned, especially with our 3D printing aspect, it's something that like we can definitely get published, and well, I mean, hopefully get published.
Well, that's our goal.
And it's very likely that we will. So I think it's definitely worth trying for
Okay.
such grants.
I agree with you. I think there are special programs out there strictly for students. This project would be a slam dunk, I think, in those special student projects, because someone can visualize it, they can understand a team of students working on it. I mean, not they understand the technology or the science or engineering behind it, but it's something you can visualize, and that sells to a panel when you can visualize something. So search around and look, see what, and just have to search and call people and ask. If you find a program manager at any of these agencies that doesn't have something specific for students, nothing beats a telephone call. Call that person up and say, listen, this is what we're looking for. I know your program might not have that, but where can you direct me? And They're sometimes hard to find, but it's amazing what a phone call will do for you. So don't hesitate to do that. But I think it sounds like you're in good shape. You have a lot of resources already available to you apparently, which is a great place to be. Switching gears a little bit, this is usually the area that Dr. Skindle handles. She was feeling really ill and she left a little while ago, so she's sorry she couldn't be here. You have 13 people, it's a big group,
Yeah.
right? Well, most of them here. You have, I love that little flow chart of your sub-teams, you know. It looked like absolute chaos to me. Okay, phase 1A, phase 1B, phase 1C, and they're all gonna come down and clash together at phase 2. There's gonna be a lot of separate moving parts, a lot of separate moving groups. Group 3 is, group 1 and 2 and 4 are totally dependent on group 3 being successful. It's all gonna come together at the end into something that's hopefully wonderful. How are you all gonna arrange to keep yourself accountable to each other? Make sure that each subgroup's making the progress according to the progress schedule they need to keep, to make sure when something catastrophic happens, which will, you have a plan for regrouping and recovering and replanning and moving forward. How's that all gonna work out? How do you plan on tackling that? This isn't gonna work how you spelled it out, it never does. There's gonna be bumps in the road. So how are you gonna do that when team 2 totally crashes and burns? What are you gonna do? I don't know who's on sub-team 2, but I picked on you anyway. So how are you gonna keep yourself accountable, make sure you progress as a team?
I don't think any, like, the sub-teams are gonna be, like, isolated from each other, really. Like, I think there will definitely be a lot of cross-interaction. So I think if, like, suppose one of the team were to completely fail in whatever that objective was, I think we would all kind of, like, regroup and see what else can we do with this project, like what other direction could we take, maybe that was novel. I think the 3D printing is something that's really new, and it's, like Dr. Bentley said, it's a pretty blank slate. So that's something that we're definitely kind of a little unsure about, but we're gonna try to make it work. But I think the integration of sensors is something we'll have a lot of people working on, so that we can at least try to make that be successful, even if our method of fabrication is something that's already been established. We have the new aspect of the sensors.
Okay.
So can I interject there? So let's say team 1 is doing soft lithography, which presumably is a slam dunk, but it's not gonna be. And you come up with a schedule so that by March 30th, you're supposed to have this done. And then, it's looking, so one of team 1A says, yeah, I think we're there. And the other person, team 1A, says, no, we're not, we're far from there. So the definition of what is done and what is right varies depending on who it is. How do you deal with that?
I think from the get-go, we kind of have to outline exactly what results we want to see that would constitute it being done, and kind of highlighting what the overall objective is and being very, very clear and detailed about that, and kind of do it before we start that phase so everybody's on the same page. And that's not necessarily something that only that sub-team would decide, it would be something we would decide as a team, because at the end of the day, all the other phases kind of rely on each other.
So when I took like world history way back before when you guys, before even, I think it was high school or something.
It's a different world.
Yeah. Poland had a diet, I think it was called, where they had actually you had to have a unanimous decision in order to have something pass. And is that what you have to have? A unanimous consent? Or how is it that so you said we're all going to agree?
I mean, we are
I mean,
a
you
team.
are a team, but does everybody have to agree?
So, go ahead, sorry. Yeah, so the way we've been doing it thus far is kind of not necessarily unanimous, like from the get-go, but we definitely talk about everyone's ideas, and we make sure to listen to what each person has to say, particularly in the cases where some people don't talk as much and have very good ideas to contribute. And like after hearing everyone's opinions on it, we'd be sure to discuss what each option could bring for us, what would be the best way to go, and then kind of through that discussion, then decide together. And then, I mean, of course, here and there might be some differing opinions, but then eventually we do try and talk about it a lot in order to come to such a conclusion that we're going to be taking this path and it's going to be the best for us.
Yeah, I've witnessed that, it's amazing to me. And in part, it's like when they sit around, they normally sit with each one with their computer, and they actually talk through their computer. Google Docs. Bizarre. So actually, I want to echo what he just said about the phone call. So instead of an email, so whoever that's for or whatever, instead of an email, delete. When you're talking to someone, the passion comes through. It's not in an email. So I really would encourage you to do that with a phone. I know you have phones, and I know you never use them to talk. But you can talk on phones. I suggest you do that.
Yep. Agency program managers, my singular opinion, are typically bored to death with their jobs. And they would love to spend half an hour talking to a bunch of energetic young researchers about an idea. So,
Yeah, and I think this kind of project, I think a lot of program managers would be caught off guard by the fact that as sophomores, you're trying to execute this over the next couple of years. I mean, it's not just like, oh, it's my summer project or something. I mean, Gemstone in general is a very special program, as obviously you all know, but I think externally, for people who aren't familiar with it, it's even more impressive. So I would agree with that. Definitely feel free to make some phone calls. A lot of these people, were professors and they took this role for a couple of years, and I think maybe not getting to have those kind of student interactions that they might have gotten used to. For certain professors who really enjoy that part of their job, it can be disappointing. So that might brighten their day. So.
okay. Finished here? Do you guys have about a minute and a half to ask us, any of us, probably not me, but a question you want to get answered? Put us on the spot.
Well, I don't know if this is even realistic, but if we wanted to think about getting patented, is that something we should start thinking about now? Do we need to be careful what we put out there?
You don't want to put too much information out there. You want to keep that to yourself. You can talk in generalities, you can talk in concepts. When you think you have something that is unique and patentable, that's when you start having a conversation through your mentor with the university about we're gonna negotiate ownership rights essentially. And that is something that will be handled through the technical side of the project. So Gemstone won't be involved in that. But it's between the mentor and some subset of the team who has the intellectual contribution that's being used. Or the whole team if it spreads out that way. It's up to you guys. And the university has people who help make that happen. But there's a preliminary patent disclosure with provisional patent, that when you think you have that good idea, you can kind of lock your idea down before you even know a lot of the details. And that's, think that way.
The big thing is just don't give an oral presentation on what you're doing until you have that provisional patent. Even if it's for a class project, that can actually make you ineligible to get an international patent. So we have a lot of issues for design day, if you've been there. If students present their projects, it's no longer available for an international patent.
Okay, so your next big step would be at the junior colloquia, right? If you have something that's, rock star quality, you want to make that provisional filed before that, which is next fall, November-ish. If not, just be careful what you present. Any other follow-ups? I think you guys did a great job. This is the jumping off point, okay? Your project is well thought through, well planned. I think highly achievable, at least a majority of it, from what I can read and hear. I don't have any concerns you're going to be successful. I think a 13-member team is hard to manage. I think you're going to have some challenges there. We are here, Scandal and I are here to help you with that. Please come see us with those things. If you come to ask me questions about where should I put the gold electrode on the side or the bottom, don't do that. Okay? I'm like, that's what these guys are here for. You just met probably two tremendously new resources for you. Don't let that relationship wither. Okay, keep it going. You're starting your professional network right here. And I think you're in good shape. I think you'll be going gangbusters as you go down the road. So congratulations.
Very good.
Yeah, great, good.
Made a lot of progress since Wednesday night.
Or not.